How drugs act. So, general principles. The emergence of pharmacology as a science came when the emphasis shifted from the describing what the drugs do to explaining how they work. In this chapter, okay, we it's set out some general principles underlying the interaction of drugs with living system. The interaction between drugs and cells is described, followed by a more detailed examination of different types of drug receptor interaction. So we are still far from the holy grail of being able to predict the pharmacological effects of a novel chemical substance or to design ab initio a chemical to produce a specific therapeutic effect. Nevertheless, we can identify some important general principles, which is our purpose in this chapter. So, Paul Ehrlich came up with something like when ligand is bound to a receptor, it shows some action. So, there's some vital forces involved in it so for the drug action to take place, and so there's an examples like 1 drop of a solution of a drug at uh, only 10 to the power of minus 10 mole still contains about 3 into 10 to the power of 9 drug molecules. So there uh, is no mystery in the fact that it may produce an obvious pharmacological response. Some bacterial toxin like diphtheria toxin act with such precision that a single molecule taken up by a target cell is sufficient to kill it. One of the basic tenets of pharmacology is that drug molecules must exert some chemical influence on one or more cell constituents in order to produce a pharmacological response. In other words, drug molecules must get so close to these constituent cellular molecules that the two interact chemically in such a way that the function of later is altered. Of of course, the molecules in the organism vastly outnumber the drug molecules, and if the drug molecules were merely distributed at random, the chance of interaction with any particular class of cellular molecule would be negligible. So, okay, he summed the drug molecule must be bound to particular constituents of cells and tissues in order to produce an effect. So, Ehrlich had summed it as corpora non agent nexi fixata. So there are some exceptions to this, like osmotic diuretic, osmotic purgatives, antacids, heavy metal chelating agents. So these are some of the exceptions to this Paul Ehrlich's dictum. So these critical binding sites are often referred to as drug targets, an obvious allusion to Ehrlich's famous phrase "magic bullets," describing the potential of antimicrobial drugs. The mechanism by which the uh, association of a drug molecule with its target leads to a physiological response constitute the major thrust of pharmacological research. Most drugs drug targets are protein molecules. Even general anesthetics, which were long thought to produce these their effects by an interaction with membrane uh, lipid, now appear to interact mainly with membrane proteins. So talking about this membrane proteins and membrane lipids. So you have the integral proteins. So integral proteins attached to the lipid bilayer. So these proteins stick out through the lipid bilayer and somewhat like the P glycoprotein. So P glycoprotein can be considered as an example. So these membrane proteins, which are the integral uh, part of membrane lipids help in the drug interaction. So all rules need exceptions, and antimicrobial and anti-tumor drugs as well as mutagenic carcinogenic agents interact directly with DNA rather than protein. Bisphosphonates used to treat osteoporosis. Osteoporosis is a disease associated with the bone which makes the bone brittle. So these bisphosphonates bind to the calcium salts in the bone matrix, rendering it toxic to osteoclast, much like a rat poison. So okay, so rat poison. So I'm not sure about the action of the rat poison. So there are also exceptions among the new generation of biopharmaceuticals that include nucleic acid, protein, and antibodies. So I'll be just detailing a diagrammatic representation of how a receptor and ligand interact. And so, okay, okay. So this is just a demonstration of how this receptor and proteins are. So this is the lipid bilayer, and so so you can call this a receptor. So through this lipid bilayer, you have the diffusion taking place. So also you have something called the this is a ligand, and there are also some proteins attached to here. So you call that okay. There are two kinds of proteins. Okay, there are proteins here. There is another protein. Okay, there is something like this. Another protein. Okay, so this is integral. So this in the red color make up the integral and peripheral proteins. So these proteins are attached to the lipid bilayer, which mainly help in what you call as association of external agents with the cell. So this okay, so mainly this is Ligand. So you call a ligand. So ligand can be like a drug. Ligand, and this one is a receptor. So this is a receptor. So okay, this one. Okay, this one is a receptor, and this is a ligand, and this is a lipid bilayer. So the association of molecules with the lipid bilayer happens like this. So as described earlier, so you have the diphtheria toxin. So diphtheria toxin can all minute quantity of diphtheria toxin is enough to elicit an action. So similarly, you have this uh, osteoporosis. So osteoporosis. Those are some of the exceptions in drug actions, how drugs act, and also there are some exceptions like osmotic diuretic antacids and heavy metal chelating agents. So this is a diagrammatic illusion of how this uh, ligand receptor action takes place. So this one mainly binds here, over here, and this channel opens up, and the channel over here opens up through modulation, and the drug elicits this action. Okay, thank you.